Good afternoon, everybody. The topic from Semester 2 Botany Honours and Paper 3 Semester 3 Mycology and Plant Pathology. That is Puccinia graminis. Puccinia graminis is one of the plant pathogen and causes a heavy loss in the wheat, which is one of the staple food of an Asian country. The genus includes about 3,000 species worldwide, but India only it reports 147 genus. It causes the rust disease or the black stem rust of wheat. The systematic positions, it belongs to the kingdom fungi, division Eumycota, subdivision Basidiomycotina, class Teleomycetes, order Eurydinis, family Pucciniaceae, genus Puccinia, and species Graminis. There are other species that are also there, but this specific species Graminis, it infects the wheat plant. And this is an obligate parasite, so it requires a specific host, that is a wheat plant and other secondary host for its completion of life cycle. It is heterogeneous parasites. It requires two different hosts to complete its life cycle. The first host or the primary host is a wheat, and the secondary host is the berberis or the mahonia plant, which is generally found on the hills. So the infection symptoms it shows like this on the wheat plant. So if we come to the life cycle of the pathogen Puccinia graminis, it shows or it completes life cycle in the two hosts as we have said. And in the first host or in the primary host, it forms different types of the spores. The spores are uridospores, N plus N, and it is produced in a specific structure that is known as uridosporangium, and teletospores N plus N, it is produced specialized structures teletosporangium, and the third spore is a basidiospores, is a N type or is a haploid type. And each basidiospores, uridospores and teletospores are dikaryotic cells, but teletospores are dicelled dikaryotic cells, and they obtain their nourishment from the hosteria. If we see the uridinal stage of the pathogen, the hyphae of the pathogen after infection on the primary host, it gets aggregated just below the epidermal layer of the cells, and they form a specialized structure which is known as uridosporangium. Out of this uridosporangium, the first kind of the spores is produced, which is known as uridosporangium. Now the uridosporangium produce uridospores. Now these uridospores are dicelled. It's more or less oval shaped structures, and after maturity it exerts pressure on the epidermis, finally ruptured in the form of slits through which the rusty colored uridospores are seen. Now again, these uridospores, they are capable of infecting the same host or the host that is present nearby. So that means it produces or it causes several infections at a time also. But during the changes in the environmental conditions, we see these are the symptoms we see during the infection of uridosporangium. Okay, so that's called the rusty appearance. And here it's rightly been called as the rust disease of uridosporangium of the wheat is also been called here. And after that, if you see the structures, it is more or less a oval here, and there is a stalk. It's a we have structures and binucleated structures and rusty in color. And after the iridospores, the another type of the spore, it is the teleal spore or teletospore stage. Late in the growing season, the uridospores are metamorphosed into another kind of the spore that is known as teletospores in the same uridospora or uridosporangium. And these teletospores are also dicell type, are also N plus N, dikaryotic, but having the dicelled. So this is more or less a black in color. So you can see this is the more or less a dicelled and dikaryotic hyphae structure is there. It looked like this, and during the infection of the teletospores, the symptoms on the primary host or the wheat, it looked like this is a blackish in appearance. Why? Because the outermost layer of this teletospores is much more thicker and it is opaque in order to, withstand a very heavy, undesiccated and very structure. So that's why it looks black in color. So it is also been rightly called here as a black stem rust of wheat. And it look like this. It's a dark brown with the black color. It's a stalked and a spindle shaped structure, and this is a dicell, one cell, two cell plus n cells, like dicells, and it is thick and smooth type. And after this, after the maturations of this teletospores, they will fall on the spore, or sometimes either still attached on the primary host. This teletospore germinates to form another third type of the spores known as the basidiospores. Now, how the basidiospores is formed? The tip of each hyphae swells and form a single binucleated oval urospores formed in a group, Urodinium or the Iridosaurus. They exert pressure on the epidermis, finally ruptured in the form of slits through which rusty colored iridospores are being seen. After this, the teletospores, each teletospore act as a basidium, and that basidium is known as hypobasidium, and the hypobasidium will convert into the epibasidium, and after epibasidium all the cells, which is a dicell, is there, n plus n cells undergo diploid diploidizations, and after that it undergo the meiotic cell divisions, and as a result, after meiosis they'll form a haploid cells, that is a 4 haploid cells, and here there will be segregation of the strands will be formed, and half will form the plus strand and half will obviously form the minus strand. So 50% will form the half strand, plus strand, and 50% will form the minus strands. And after that, each basidium, phragmobasidium, will form the small haploid basidiospores. Now, after the basidiospores is been formed, now these basidiospores will continue its life cycle in the secondary host. Now, these basidiospores are haploid. They may be of the plus strains or minus strains, and after liberations or disseminated from the teletospores, they will germinate on the upper epidermis of the Burberry leaf and produce another type of the spores, which is known as pycnospores, or within the pycnosporangium. So the secondary or the continuations of the secondary host will be there. So that will be dealt in the second part of the video. Thank you.